afternoon, all of you. In last lecture, I have discussed the preservation of pharmaceutical products by using antimicrobial agents. Today, we will see the next point of our syllabus, that is evaluation of microbial stability of formulation. Yes, today we will see the next point of our syllabus, that is evaluation of microbial stability of formulation. Generally, the question asked in examination likewise, what is evaluation of microbial stability of pharmaceutical product, or explain in detail evaluation of microbial stability of pharmaceutical product, or what is microbial stability of pharmaceutical product and how it is evaluated, or explain in detail challenge test for microbial stability of pharmaceutical product. The microbial stability test, it involves following steps: first, sampling; then second, growth media. In sampling, the number of containers to be used and volume to be tested are specified in the pharmacopoeia. It means it is evaluated as per pharmacopoeia. Then samples of aseptically manufactured products are collected in a beginning of, then during and end of batch fill. Here samples are collected in beginning, then during and end of batch fill, and also after any kind of interventions or stoppages. I am repeating here. See here what is important. During evaluation, the samples are collected. Sample of aseptically manufactured product are collected in beginning, during and end of batch fill, and also after any kind of interventions or stoppages. Then the samples from terminally sterilized product are collected from previously identified cool spots within load method as specified in the pharmacopoeia. Then, if the product can be filtered by the membrane filtration method is preferred, while direct inoculation is used as an alternative method. Then second, that is growth media, that is capital SCD, that is soybean casein digest medium. SCD, it means the soybean casein digest medium, and fluid thioglycolate medium, that is capital FTM, are the media are used for detection of aerobic and anaerobic organisms. These two media are used for detection of aerobic as well as anaerobic organisms. Then the efficiency of media to support growth of a range of low number of organisms in the presence of product is demonstrated by validation studies. Then the bacterial growth should occur after days, and growth of mold should occur after 5 days. Then media is either procured or made in house by validated sterilization technique. Before using this media, its growth supporting quality should be tested. Then the batch number and shelf life of the media, it should be allotted. Then second, that is incubation period, SCB, that is soybean casein digest medium, is incubated at 20 to 250 degrees Celsius, and FTM, that is fluid thioglycolate medium, this media is incubated at 30 to 350 degrees Celsius, both for 14 days in a test container, which should be periodically examined. Then negative test control and positive test control. In negative test control, before using the media, either a portion of it or the complete batch, it should be incubated. Before using the media, either a portion of it or the complete batch, it should be incubated for 14 days. Then incubation may be done simultaneously with the test. Then a negative product control atom, identical in type and packaging to actual product under test, it should be included in each test session. This makes interpretation of the test result easy. Then second, that is positive control test. With the positive test control, the bacteriostasis or the fungostasis test should be proved that The media can support the growth of a range of low number of organisms in the presence of products. Then the bacterial growth should occur after days and that of mold should occur after 5 days. So what is the result for this? The any type of growth is identified, that is genotypic, or the automated or the semi-automated system. And it should be verified from time to time by use of reference stain. So what is preservative? The preservative are the antimicrobial agent that can be found in this description. Also comes under this point, that is evaluation. The preservatives are antimicrobial agent that can be found in nearly all consumer products, including foods. The challenge test carried out by the inoculation of following microbes. First, the name of organism and description. Pseudomonas aeruginosa, it is gram negative, very hard bacterium. E. coli, it is gram negative, similar to many pathogens. Then Staphylococcus aureus, it is gram positive bacterium, opportunistic pathogen. Then Candida albicans, it is fungus or yeast, opportunistic pathogen. Then Aspergillus niger, it is fungus or mold, opportunistic pathogen. Then there are different products are classified into various categories as follows. First, that is category 4, 1, 2, 3 and 4. Description and passing criteria. In description, in category 1, the injection parenteral including emulsion. Passing criteria is bacteria. Then for optic products, no less than 1 gram 1.0 log reduction for the initial calculated count at 7 days. Then sterile and nasal product, not less than 3.0 log reduction at 14 days. Then for ophthalmic products made with aqueous base or the aqueous basis of the vehicle, no increase from the 14 days count at 28 days. And yeast and mold, no increase from the initial calculated count at 7, 14 and 28 days. Then category second, the topical product made with aqueous base of the vehicle. Then in bacteria, no less than 2.0 log reduction from the initial calculated count at, that is your 14 days. Then non-sterile nasal product, and then emulsion including those applied to only mucous membrane or applied to mucous membrane, no increase from the 14 days count at 28 days. For yeast and mold, no increase from the initial calculated count at 14 and 28 days. Then category 3, third, oral products made with aqueous base of the vehicle other than antacids. Here what I am saying, that is other than antacids bacteria, no less than 1 log reduction from the initial count at 14 days, or the no increase from the 14 days count at 28 days. And for yeast and molds, no increase from the initial calculated count, that is 14 and 28 days. And last category 4, the antacid made with aqueous bases, that is bacteria, yeast and mold, no increase from the initial calculated count, that is 14 and 28 days. Then next, that is the preservative challenge test consists of following basic steps. First, the evaluation for the presence of pathogen after manufacturing. Then second, the product is separated out into 5 separate containers as a sample for testing. It means the product is separated in different 5 separate containers as a sample for testing. Then above separated containers is inoculated with different microorganisms at high concentration in order to find out initial challenge concentration. Inoculation with different microorganisms. Then the initial concentration of each inoculated microorganism is determined by inoculating a control substance. Yes, the initial concentration of each inoculated substance, each inoculated microorganism, each inoculated microorganism is determined by use of or by inoculating a controlled substance and using a standard dilution and a plating techniques. At the start of test, 1 ml or 1 gram of the product is diluted in a volume of chemical neutralizer broth. Then the incubation period at room temperature for at least 7 days or 14, 21 or 28 days. An inoculated product is evaluated at regular interval, which is depend upon the type of product. Then after 48 hours of incubation, the surviving microorganisms are counted. After 48 hours of incubation, next that is the surviving microorganisms are counted, and the log reduction of each microorganism at each interval is reported by the passing criteria. And last, the product is re-evaluated for the presence of the microorganism that were inoculated into it, which depend upon the shelf life of product. So this is all about the evaluation of microbial stability of pharmaceutical products.